Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, today in the continuation of the chapter that is molecular basis of inheritance, we are going to study one of the interesting topic that is regulation of gene expression. Okay, dear students, see there are approximately 30,000 near about genes are there in human DNA, which is a 2.2-meter long. So there are so many genes in the genome of every individual or every species, and when the product of particular gene is required, at that time that particular gene is getting expressed and forming a protein. But if it is not required, it cannot express and it do not form a protein. So such a regulation is there that according to the need, the gene are getting expressed and not expressed, or they are forming a protein or not. Okay, that is there. So we need to understand that how this regulation occur actually at a gene level. So if we talk about the regulation of gene expression means protein formation. So the regulation of gene expression refers to a very wide sequence of events that may occur at various levels. Means it is a very broad term and it occur at a various level. And we consider here that gene expression means it result in the formation of polypeptide. So considering that the gene expression result in the formation of polypeptide, the polypeptide synthesis will be regulated, or it can occur or it cannot occur, and it can be regulated at a several level. In eukaryote, dear student, if we talk, regulation can be exerted at several levels. Okay, and it is then known that where we can decide that polyprotein will form or not in eukaryotes. Then it can occur at a transcription level, means formation of primary transcript. There the regulation can occur. Either the primary transcript can form or not. Same way, this regulation can occur at a processing level, or a regulation of splicing we can say, which is a part of post transcriptional modification in eukaryote. If it does not occur, then also the further protein synthesis cannot occur. And after a formation of mRNA, if regulation is also occurring, if splicing also occur and mRNA is formed, mRNA transport occur from nucleus to the cytoplasm. If this is blocked, then also the protein synthesis cannot occur. And at translation level also, due to the some mistake, it cannot occur or can occur. So all these four levels are there where the regulation can occur in case of regulation of protein synthesis or gene expression can occur in the eukaryotes. The genes in a cell are expressed to perform a particular function or a set of function. See, if we talk about genes in a cell, the genes express and they form a particular protein, and that particular protein will be responsible for a particular phenotype. So gene has the ability to code for one phenotype or a particular function and the set of functions. So see, this is the nucleus. In nucleus, the chromosomes are there. In chromosomes, DNA are there, and in DNA, these genes are located. Now we are going to talk about this example, but dear students, we remember in eukaryote, functionally related genes do not represent an operon. Functionally related genes do not represent operon. They are not side by side. They are present on the different sites on the different chromosomes can occur. Now, if we talk, let's to understand the regulation, let's take an example of this enzyme that is a beta galactosidase, and it is synthesized in the E. coli. Now, this beta galactosidase is known to catalyze the hydrolysis of the disaccharide, that is lactose into galactose and glucose. Lactose is divided into galactose and glucose by this enzyme beta galactosidase. And during breakdown, the energy is released that bacteria use them, and further bacteria use them galactose and glucose as a source of energy. Now, this galactose is there, lactose is there, which is also known as galactose beta 1,4 glucose. By beta 1,4 linkage, they are joined and they break by the beta galactosidase into galactose and the glucose, and the structure are given here. Hence, if the bacteria do not have a lactose, around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta galactosidase, because beta galactosidase enzyme is required for the breakdown of this lactose. But if lactose is not there, then that the functionality of this enzyme is not that required. So this enzyme is not going to be synthesized. In a prokaryote, if we talk about this regulation, such a regulation, then in prokaryote it occur at only one point. And in prokaryote, control of the rate of transcription initiation, if we talk, control of the rate of transcription initiation is the predominant target site for control of gene expression. So in prokaryote, transcription initiation is the main site where the regulation can occur. See, this is the particular DNA. In DNA, if mRNA is forming, the transcription is occurring in this way. Okay, and mRNA is forming, and this is the promoter region. Okay, this is the particularly sense strand is red one, and antisense or nonsense strand we can say this 3 days to by. This is a template strand. Sorry, antisense is this one. Green color is a antisense or template strand, and sense strand or coding strand is given in a red color. And the mRNA synthesis occurs. So at a transcription initiation, this is going to occur. Now in a transcription unit, we know that the activity of RNA polymerase at a given promoter is interregulated by interaction with the accessory protein. RNA polymerase interact at a promoter region, and the interaction occur between with the accessory proteins, and this accessory proteins helps to recognize a particular start site by the RNA polymerase. So when RNA polymerase bind with the promoter region, the regulatory proteins are there which can act both positively activator or inhibitor. So we are going to discuss that how this activator are produced and how they work. So the accessibility of the promoter region of a prokaryotic DNA is in many cases regulated by the interaction of protein with the sequence termed operator. See, in the operon, now to understand this regulation of gene expression, operon model is given. Now in operon model, certain genes are there, and these certain genes are present in a particular sequence. They have some regulatory genes, okay. Then the product of this regulatory gene bind with the operator. Promoter is a different gene where the RNA polymerase interact, and there are different structural genes. So operator genes are the genes where the promoter accessibility for the RNA polymerase is regulated by the interaction of some proteins with the sequence termed promoters, operators. So this is the operator region. So the operator region is adjacent to the promoter element in most operon, and in most cases the sequence of operon bind a repressor protein. So this repressor protein is going to bind with this operator region. How? Let's discuss. We will understand in this later part. Now, if we talk about each operon has a specific operator and specific repressor regions. If we talk about the operon is nothing but what group of genes whose functions are coordinated. So here you can see these are the some of the genes like regulatory gene is there. This is the LECI, then this is the promoter region, then this is the operator region, and this is the what structural genes. So each operon has a specific operator and the specific repressor genes. And for example, LEC operator is present only in LEC operon, and it interacts specifically with the LEC repressor only. So here you can see this is the operator region, okay, and this is the regulatory gene. The product of regulatory gene is going to interact with the operator region. Yes or no? And this is the promoter region where RNA polymerase is going to bind. So this is the operator, and specific repressor is going to be a product of this regulatory gene. To understand that, we need to understand the LEC operon. Before that, let's talk what type of genes are present in this operon and what is operon. Now, if we talk about operon, first discuss what is operon concept. Now, if we talk about operon concept, Then it is known that Francis Jacob, a geneticist, and Jacques Monod. So Jacques Monod and Francis Jacob. Jacques Monod was the biochemist and Jacob was the geneticist. They proposed model of gene regulation. Model of gene regulation known as operon model in bacteria. Now, what is operon then? So, operon is nothing but operon is a coordinated group of genes. It is a coordinated group of genes such as structural gene, operator gene, promoter gene, regulator gene. So such genes are coordinated genes, and this group of coordinated genes is known as operon. Now this function together and regulate metabolic pathway as a unit. Okay, and if we take about the different examples of operon, then there are several operon like lac operon, which deals with the metabolism of lactose. Then tripoperon. Tripeperon means tryptophase operon. Era means arabinose operon. Then histidine operon, his operon. Valine operon, VAL, etc. Such operon are known. And they have a regulator gene. They all have number one, regulator gene. This regulatory gene synthesizes a biochemical or regulatory protein. It synthesizes a regulatory protein or a biochemical which act positively as activator or negatively as repressor. And it is known that it controls the activity of operator gene. This regulator gene control the activity of operator gene. Okay. So after regulatory gene, next we are going to discuss the second type of gene, that is operator gene. Now, if we talk about operator gene, it is a gene. So, if we talk about this operator gene, this gene receive the product of regulatory gene. This gene receives a product of regulatory gene, and it is known that it allows the functioning of operon. It allows the functioning of operon when it is not covered by, it is not covered by product of regulatory gene. Means if product of regulatory gene is repressor, if repressor bind with the operator, the gene of operator will not allow the further transcription of the further structural genes. Third is promoter region or promoter gene. It provide attachment site for RNA polymerase. And fourth is structural gene which transcribe mRNA for polypeptide synthesis. Okay, such a way this all are actually arranged and coordinated. Now to understand this, how they are particularly functioning, if we discuss one operon, then it will be easy for us to discuss this. So let us discuss for the understanding of this regulation or operon concept in a very defined manner, lac operon, which is going to describe the lactose metabolism. And here lac refers what? Lactose. So the elucidation of the lac operon was also a result of a close association between the geneticist Francis Jacob and the Jacques Monod. This is the two scientists who worked on this lac operon. The scientists were the first to elucidate a transcriptionally regulated system. And in lac operon, here lac refers to the lactose. Now this lac operon, if we talk, here a polycistronic structural gene are there. Means they are going to synthesize a polycistronic mRNA, and polycistric mRNA have a information for a many polypeptide or many enzymes or many proteins. And if the genes are a polycistronic genes are for a polycistronic mRNA, then polycistronic structural gene it is known as, and it is regulated by a common. So multiple structural genes are regulated by a common promoter and regulatory gene, then it is known as polycistronic structural gene. Here you can see this is the arrangement of gene, and such arrangement is very common in bacteria and is referred as an operon. Now in this operon you can see here this is the regulatory gene. Okay, and it's this promoter. This is the operator gene. This is the promoter gene. And this is the structural gene. Now structural gene are actually going to code for the enzymes. And here three genes are there. This common promoter for three genes and common operator. So these three genes are known as going to form a one common mRNA. And that mRNA is known as a polycistronic mRNA. And these genes are known as polycistronic genes. Now we remember the product of this regulatory gene is going to bind with this operator gene. Okay, and this arrangement of gene is known as operon. A few such example if we take, then lac operon is there, trip operon, that his operon, era operon, valine operon, that all we discussed. In most other operon as well as the genes present in the operon are needed together to function in same or related metabolic pathway. Lactose is the substrate for the enzyme beta-galactosidase, we know, and it is regulatory switching on and off the operon. It shows the switch on switch off mechanism. So lactose is termed as an inducer here. Now see, this in case of the absence of particularly we can say a regulator. Dear students, be remember this is the promoter, this is the I gene, this is the promoter, this is the operator, this is Z, Y, A are the particular structural gene. Now here it occurs in the absence of inducers. Be remember this is the leg Z gene. This leg Z gene code for the enzyme beta galactosidase which carry out a breakdown of beta galactoside or lactose. And this beta galactosidase is mainly responsible for the hydrolysis of the lactose into monomeric units, that is the galactose and the glucose. Whereas lac Y, if we talk, it code for the enzyme permease. And permease increase the permeability of a cell for the lactose or beta galactosides. Whereas lac A, if we talk, it code for the trans-acetylase enzyme. And this trans-acetylase enzyme transfer acetyl group to the lactose and make it stable. Now, when the inducer is there, in the inducer is absent, see, this I gene is a regulatory gene. And this is the promoter for the regulatory gene. Now, this regulatory gene is the I gene, which is a housekeeping gene, which is going to synthesize its protein 24/7. So, it is going to form its repressor mRNA and it is going to synthesize the repressor molecule. Now, this repressor, which is a product of regulatory gene, it binds with the operator gene. And when it binds with the operator gene, it does not allow RNA polymerase to interact with promoter and transcribe this gene further. Okay, so when operator gene is there, this RNA polymerase, if we talk, which bind with this promoter region, okay, that RNA polymerase is not going to bind with this promoter region and not going to transcribe the structural genes. So here repressor binds with the operator region and prevents the RNA polymerase from transcribe the operon or a structural gene. Then in the absence of inducer, the structural gene are not going to be transcribed. So this condition is known as switch off condition for the operon. Now if you want to make a product, means these genes are not going to express. If we want to make an expression of this, then we need to convert this switch off operon into the switch on. And to convert switch off into the switch on, so this occur in the particularly switch off condition. Now if we want to discuss in the presence of this inducer, that is the switch on condition, then in the presence of inducer, what will happen? This inducer, this yellow colored inducer is going to bind with this repressor and it does not allow this repressor to interact with the operator. And if it does not allow the interaction of this with the operator, the RNA polymerase will interact with the promoter and transcribe the further gene. So the leg Z, leg Y, leg A, the common mRNA will be made, and this is a polycistronic mRNA. Now this polycistronic mRNA will form a leg Z gene form beta galactosidase enzyme, leg Y gene code for permease enzyme, and leg A will code for the trans-acetylase enzyme in the presence of inducer. Then it will be called as a switch on condition. Means structural gene are getting expressed. So this way the regulation is going to take place. Now after this switch on and switch off condition understanding, if inducer is there, then it is a switch on condition, and in the absence of inducer it is a switch off condition. So here if we discuss this, then it is known that the regulation, if we talk here, then it is inducible operon. It is inducible operon, and here inducer is required. The inducer can be allolactose or lactose. So when lactose or allolactose is there, it is going to bind with the repressor and allowing the synthesis of the structural gene, and beta galactosidase enzyme will come and that will cleave the lactose. Such a way this operon goes on particularly working. Now here, if we talk about regulation of lac operon, regulation of lac operon can be negatively or positively. Regulation of lac operon is positive with respect to inducer because it make it switch on, and negative with respect to repressor because it make it switch off. So it is under a positive as well as negative regulation. Okay, so this is all about the particularly the lac operon. So what we understood by understanding this lac operon, that in the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, it is transported into the cell through the action of permease enzyme. Remember, a low level of expression of lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cell. If somewhat permease is there, then only it can allow the lactose into the cell. Now, the lactose that induces the operon in a following manner. The repressor of the operon is synthesized all the time, hence said to be constitutively from the I gene. The repressor protein bind with the operator and prevent a RNA polymerase from transcribing the operon. So it is not allowing. In the presence of inducer, such as lactose and allolactose, when it binds with the repressor, it inactivates repressor and thus it does not allow bind to repressor with the operator gene. So it allows the RNA polymerase to access the promoter and transcription process. Essentially, regulation of lac operon can be visualized as a regulation of enzyme synthesis by its substrate. It can be visualized as a regulation of enzyme synthesis by its substrate. Means lactose is helping the lac Z, lac Y, lac A gene to express because it is acting as a inducer and not allowing repressor to bind with the operator. So remember, glucose and galactose cannot act as an inducer for operon. Lactose is going to act or allolactose is going to act. And regulation of lac operon by repressor is preferred as a negative regulation, whereas lac operon is under the control of positive regulation as well as, but it is beyond the scope of discussion at this level. Okay, so we mainly, if ask it is which type of regulation, then you will answer it is a negative regulation. Okay, so this is about the particularly we can say lac operon. Now we will discuss a trip operon as well as, okay, but that we will discuss in next lecture. Let's discuss some of the MCQs. An enzyme called beta galactosidase is synthesized by, obviously it is synthesized by E. coli. The lac operon model is proposed by, obvious we have discussed, it is by the Jacob and Monod. Lac ZY gene code for the, it is code for beta galactosidase. Okay, so this is all about the what lac operon and now what is regulation of gene expression. Now trip operon is remaining, that we will discuss in a separate lecture. Okay, thank you.